Hello everyone, welcome back to my channel. So today we are going to discuss the most important questions of the chapter Biotechnology, Principles and Processes. So these questions are important for the biology class 12 term especially. So the first most important question is, first part, while cloning vectors, which of the two will be preferred by biotechnologists, bacteriophages or plasmids? And you have to justify your answer. Biotechnologists prefer bacteriophages for the cloning over plasmids because they have very high copy numbers of their genome within the bacterial cells, whereas some plasmids may have only 1 or 2 copies per cell and others may have 15 to 100 copies per cell. The second part of the question is, name the first transgenic cow developed and state the improvement in the quality product produced by it. This question has been asked in the CBSE Simple Paper 2018-19. Answer is, transgenic cow Rosie produced human protein enriched milk, which was 2.4 grams per liter. The second question is, what are the two core techniques that enable the birth of biotechnology? The two cores are, first, genetic engineering techniques to alter the chemistry of genetic material, which is DNA and RNA, to introduce these into host organisms and thus change the phenotype of the host organism. Second core reason can be maintenance of sterile ambience and chemical engineering processes to enable the growth of only desired microbe eukaryotic cell in large quantities for the manufacture of biotechnological products. The question 4. What does EcoRI signify? How its name is derived? So EcoRI signifies the name of a restriction endonuclease, and then you have to write some rules for the nomenclature. Question 5. What are recognition sequences or recognition sites? This is also a very important question. The sites recognized by the restriction endonucleases are recognition sites. The recognition sequences are different and specific for the different restriction endonucleases, and these sequences are palindromic in nature. Question 6. Define vector. Give the properties of a good vector. Again, a very important question. A vector is a DNA molecule that has the ability to replicate in an appropriate host cell and into which the DNA fragment to clone is integrated for cloning. A good vector must have the following properties, and you have to write some properties for the vector, like it should have an origin of replication, it should be easy to isolate and purify, and it should get easily introduced into the host cells. Question 9. Explain the principle that helps in separation of DNA fragments in gel electrophoresis. Again, it is asked in CBSE 2009. The answer is, gel electrophoresis is a technique of molecules such as DNA, RNA, protein on the basis of their size under the influence of the electric field so that they migrate in the direction of the electrode bearing the opposite charge. Positively charged molecules move towards cathode and vice versa. These molecules move through a medium or matrix and can be separated on the basis of their size. In this question, you can also make a proper diagram. Question 10. Give the applications of PCR technology. Again asked in CBSE 2013. So the applications are the amplification of DNA and RNA. Second, determination of orientation and location restriction fragments relative to one another. Third, detection of genetic diseases such as sickle cell anemia, phenylketonuria, and muscular dystrophy. Question 11. Why is Agrobacterium-mediated genetic transformation in plants described as a natural genetic engine of plants? So the answer is, Agrobacterium tumefaciens is a plant pathogenic bacterium which can transfer part of its plasmid DNA because it infects host plant, and you can explain the proper procedure. Question 12. Differentiate between gene therapy and gene cloning. The difference between gene therapy and gene cloning is that the gene therapy is the replacement, or we can say it is the alteration of defective genes responsible for the hereditary diseases by normal genes. Cloning is the technique of obtaining identical copies of a particular segment or. Question 13. From what you have learned, can you tell whether enzymes are bigger or DNA in molecular size, and how did you know? The answer is, DNA molecules are bigger in size as compared to the size of enzymes because the enzymes are proteins, and protein synthesis occur from a small portion of DNA called genes. Thank you so much for watching my video, and if you like my efforts, please subscribe to my channel and you can comment down which chapter you want for the next video. Thank you.